Genomics is a study of entire genomes, including the complete set of genes, their nucleotide sequence and organization, and the interactions within species and with other species. Genome mapping is the process of finding the location of genes on each chromosome. The maps that genome mapping create are comparable to maps we use to navigate streets. A genetic map is an illustration that lists genes and their location on a chromosome. Genetic maps provide a big picture similar to an interstate highway map and use genetic markers similar to a landmark. A genetic marker is a gene or sequence of a chromosome that co-segregates, shows linkage with a specific trait. Early geneticists called these linkage analysis. Physical maps present the intimate details of smaller chromosome regions, similar to a detailed road map. A physical map is a representation of the physical distance in nucleotides between genes and genetic markers. Both linkage maps and physical maps are required to build a genome's complete picture. Having a complete genome map of the genome makes it easier for researchers to study individual genes. Human genome maps help researchers in their efforts to identify human disease-causing genes related to illnesses like cancer, heart disease, cystic fibrosis. We can use genome mapping in a variety of applications, such as using live microbes to clean up pollution or even prevent pollution. Research involving plant genome mapping may lead to producing higher crop yields or developing plants that are better adapt to climate. The study of genetic maps begins with linkage analysis, a procedure that analyzes the recombination frequency between genes to determine if they are linked or show independent assortment. Scientists used the term linkage before the discovery of DNA. Early geneticists relied on observing phenotypic changes to understand an organism's genotype. Shortly after Gregor Mendel, the father of modern genetics, proposed that traits were determined by what we now call genes, other researchers observed that different traits were often inherited together and thereby deduced that the genes were physically linked by their location on the same chromosome. Gene mapping relative to each other based on linkage analysis led to developing the first genetic maps. Observations that certain traits are always linked and certain other traits are not linked came from the studying of offspring of crosses between parents with different traits. For example, in garden pea experiments, researchers discovered that the flower's color and the plant pollen shape were linked traits, and therefore the genes encoding these traits were in close proximity on the same chromosome. We call exchanging DNA between homologous chromosome pairs genetic recombination, which occurs by crossing over DNA between non-homologous chromosomes, such as non-sister chromatids. Linkage analysis involves studying the recombination frequency between any two genes. The greater the distance between the two genes, the higher the chance that the recombination event will occur between them, and the higher recombination frequency between them. Although there have been significant advances in medical sciences in recent years, doctors are still confounded by some diseases, and they're using whole genome sequencing to discover the root of the problem. Whole genome sequencing is a process that determines an entire genome's DNA sequence. Whole genome sequencing is a brute force approach to problem solving when there is a genetic basis at the core of a disease. Several laboratories now provide services to sequence, analyze, and interpret entire genomes. The basic sequencing technique used in all modern-day sequencing projects is the chain termination method, also known as the dideoxy method, which Fred Sanger developed in the 1970s. The chain termination method involves DNA replication of a single-stranded template by using a primer and regular deoxynucleotide, which is the monomer or a single DNA unit. The primer and the deoxynucleotide mixed with a small proportion of fluorescently labeled dideoxynucleotides. The dideoxynucleotides are monomers that are missing a hydroxyl group at the site which another nucleotide usually attaches to form a chain. Scientists label each dideoxynucleotide with a different color fluorophore. Each time the dideoxynucleotide incorporates into a growing complementary strand, it terminates the DNA process, which results in multiple short strands of replicated DNA that are each terminated at a different point during replication. When gel electrophoresis processes the reaction mixture after separating into a single strand, the multiple newly replicated DNA strands form a ladder because of the different sizes. Because the dinucleotides are fluorescently labeled, each band on the gel reflects the DNA strand size and the dideoxynucleotide that terminated the reaction. The different colors of the fluorophore labeled dideoxynucleotides help identify the dideoxynucleotide incorporated in that position. Reading the gel on the basis of each band's color on the ladder produces a template strand sequence. DNA microarrays are methods that scientists use to detect gene expression by analyzing different DNA fragments that are fixed to a glass slide or silicon chip to identify active genes and sequences. We can discover almost 1 million genotypic abnormalities using microarrays, whereas whole genome sequencing can provide information about all 6 billion bases in the human genome. Although studying genome sequencing medical application is interesting, this discipline dwells on abnormal gene function. Knowing about the entire genome will allow researchers to discover future onset diseases and other genetic disorders early. This will allow for more informed decisions about lifestyle, medication, and having children. Genomics is still in its infancy, although someday it may become routine to use the whole genome sequencing to screen every newborn to detect genetic abnormalities. In addition to disease and medicine, genomics can contribute to developing new novel enzymes that convert biomass to biofuel, which results in higher crop and fuel production and lower consumer costs. This knowledge should allow better methods of control over microbes that industry uses to produce biofuels. Genomics could also improve monitoring methods that measure the impact of pollutants on ecosystems and help clean up environmental contaminants. Genomics has aided in developing agrochemicals and pharmaceuticals that could benefit medical science and agriculture. Pharmacogenomics, or toxic genomics, involves evaluating drug effectiveness and safety on the basis of information from an individual's genomic sequence. We can study genomic responses to drugs using experimental animals, such as rats or mice, or live cells in a laboratory before embarking on studies with humans. Studying changes in gene expression could provide information about transcription profile of the drug's presence, which we can use as an early indicator of the potential for toxic effects. For example, genes involved in cellular growth and controlled cell death, when disturbed, could lead to cancerous cell growth. Genome-wide studies can also help find new genes involved in drug toxicity. Medical professionals can use personal genomic sequence information to prescribe medications that will be most effective and least toxic on the basis of an individual's patient's genotype. The gene signatures may not be completely accurate, but medical professionals can test them further before pathological symptoms arise. Traditionally, scholars have taught microbiology with the view that it is best to study microorganisms under pure culture condition. This involves isolating a single cell type and culturing it in a laboratory. Because microorganisms can go through several generations in a matter of hours, their gene expression profiles adapt to the new laboratory environment very quickly. In addition, the vast majority of bacterial species resist culturing in isolation. Most microorganisms do not live as isolated entities, but in microbial communities or biofilms. For all these reasons, pure culture is not always the best way to study microorganisms. Metagenomics is the study of the collective genomes of multiple species that grow and interact in an environmental niche. Metagenomics can be used to identify new species more rapidly and to analyze the effects of pollutants on the environment.